Good evening, friends. Today I am going to discuss some question about medical lab technology, and the question includes topics like biochemistry, hematology, microbiology, and pathology. As you can see, I hope you will enjoy this live stream. So I have came live, and if you have any question, then you can ask me. And please like and subscribe my channel. Now I will start. Okay, now can I start? So here the first question is given as uh, what is the anticoagulant of choice for complete blood count? So it is a question of hematology. Okay, and for complete blood count we should need a sample which have not gone under hemolysis. So we know that to protect the morphology of the red blood cells, we have to take the sample of uh, CBC, means complete. If we want to do CBC, then we have to uh, keep the morphology of the sample cells intact. So in order to uh, in order to save the morphology of the cell, we have to use EDTA. Okay, it is EDTA. Okay, let me. Why is the eraser? It is EDTA. Okay, it is EDTA. Now I will come to the next question. So can you tell me which is the universal red cell donor? Anyone? Universal red cell donor. Anyone who can say me which one is the universal red cell donor? Universal red cell donor is, I think, mm, it is O positive. Okay, it is O positive. Okay, Of which blood group is considered the universal red cell donor? It is O positive, because in blood group O, neither A antigen is present and neither B antigen is present. So it can donate blood, since A antigen and B antigen both are absent. Therefore, it can donate blood to anyone. Okay. Now next question. Here the question has been asked: in blood group determination, why is tube method generally considered more reliable than the slide method? The answer will be option number B. That is, it allows for centrifugation and detects weaker agglutination reactions. Okay, because in tube method we can centrifuge the sample. Therefore, it detects the weaker agglutination reactions, which is not, which is impossible to detect in slide method. So, tube method is generally considered more reliable than slide method. Now, next question. The normal lifespan of a red blood cell in circulation is approximately, and the answer of this question will be 120 days. Okay, 120 days. Now, which cell is primarily helpful for production of antibodies? Here the question comes: which cell is primarily helpful in production of antibodies? Production of antibodies. Which cell is responsible? Which cell is responsible for production of antibodies? It will be lymphocyte. The answer of this question will be lymphocyte. Okay, which cell is responsible for production of antibodies? It will be lymphocyte. Now we will come to the next question, and it is given that what is the optimal storage of packed red blood cell? Optimal storage, STORAGE, optimal storage of packed red blood cells. It will be 1 degree centigrade to 6 degree centigrade. The answer of this question will be 1 degree centigrade to 6 degree centigrade. Okay. Now the next question is: a high eosinophil count is most commonly associated with high eosinophil count. High eosinophil means it is if we have high eosinophil count, then we can say that it is due to parasitic infection and allergic reaction. I think you are getting interested in it. Now, the next question comes as which strain is most commonly used for peripheral blood smear? It will be Lisbon strain. The answer of this question will be Lisbon strain. We use Lisbon strain during peripheral blood smear. You have to remember it. Now, reverse blood grouping involves testing the patient, testing the patient's serum against known red cells. Okay, in reverse blood grouping, we test the patient's serum against the known red blood cell, and in forward blood grouping, we test red cells against known antisera. Okay. Now here the question is given as what is the primary function of platelet? We know that platelet is also known as thrombocytes. So from the name itself, we can conclude that the primary function of platelet is hemostasis and blood clotting. Platelets in our body helps in blood clotting, and we have to remember it. Which of the following test is the best indicator of long-term 2 or 3 months glucose control in diabetic patient? The answer of this question will be HbA1c, which is glycosylated hemoglobin. It measures the average of 120 days hemoglobin. Okay, this test helps to measure the average of 120 days glucose present in a patient body. Here the question is given as sodium fluoride is used as a preservative. Sodium fluoride is used as a preservative in blood collection tube for glucose testing because the sodium fluoride helps us in inhibiting glycolysis. It denies glycolysis. So we have to collect blood when we want to do test of glucose or diabetes. We will always take blood in sodium fluoride tube, and it is gray in color. So the top of the tube is gray in color. So we can say that as sodium fluoride helps in inhibiting glycolysis, so we have to use the tube of sodium fluoride. Now, elevated level of blood urea nitrogen and creatinine. When someone has elevated levels means raised levels of blood, urea, nitrogen, and creatinine, then which organ is affected? In this question is given that if someone has raised level of blood, urea, nitrogen, and creatinine, then which organ is affected? The right answer of this question will be kidneys. Okay. Are you finding this question interesting? Please let me know. If you have any question, then you can ask me. I will definitely answer you. Here the question is given as which cardiac method is considered the most specific for acute myocardial infarction, that is heart attack. And the right answer of this question will be troponin. Okay. Right answer of this question will be troponin. Good cholesterol. The good cholesterol that help remove the cholesterol from the bloodstream is what does the good cholesterol help us? The good cholesterol help us to remove the low density lipoprotein, and it will be LDL. The good cholesterol means HDL help us to remove the LDL, that is low density hemoglobin. The only next question is in a gram strain, what is the primary strain used? Can anyone tell me? Yeah, in a gum straining process, what is the primary strain? What is the primary strain in a gum straining process? It is crystal violet. Okay, crystal violet will be the right answer. So crystal violet is the primary strain used in gram straining process. Standard sterilization using an autoclave requires which condition? It will be 121 degrees centigrade at 15 psi pressure for 15 to 20 minutes. Autoclave are used in microbiological lab which are operated at 121 degrees centigrade at 15 psi pressure and which operates for 15 to 20 minutes to fully sterilize the culture and medical lab equipments. Okay. Now here it is given as which organism is the positive agent of tuberculosis and requires an acid fast train Jalen Nielsen for identification. So which organism needs an acid fast training and it is a positive agent of tuberculosis for identification? It will be Mycobacterium tuberculosis. Now, McConkey agar. is an example of a medium that is. So McConkey agar help us to differentiate between lactose fermenting and non-lactose fermenting bacteria. So it is a selective and differential media. So the type of media which the McConkey agar is the selective and differential media. Now, bacteria that are spherical in shape are classified as the bacteria which are spherical in shape and is classified as cocci. Cocci are spherical in shape. Bacilli are rod shaped. Vibros are coma shaped. Okay. The most common fixative used for routine histopathology tissue sample is it will be 10% buffered formalin. Okay, it will be 10% neutral buffered formalin. If you have any question, then you can ask me. I am ready to answer you. Please like and share my videos so that I get motivated to bring more videos like this, more live stream. I can do more live stream like this. And please share to your friends because sharing is caring. Okay. What is the full form of PPE? You have listened about the PPE kits during coronavirus. Have you listened about PPE kits? So the acronym for PPE will be personal protective equipment. Okay. So what is the acronym of PPE? It is personal protective equipment. Now I will come to the next question, and here the next question is given as a microtome is an instrument used in the laboratory primarily for microtome is an instrument which is used in laboratory as why is a microtome used? It is used for cutting extremely thin section of tissue and this instrument is used in histopathology. Okay, where this instrument is used, it is used in histopathology lab. Which of the following is considered a pre-analytical error in the laboratory? Pre-analytical error. means the errors which can happen before analysis of sample, and the answer will be, it will be mislabeling a patient blood tube. Suppose you have taken patient X sample and you have written in the tube the name of the patient Y. So in that case, we can say that it is a pre-analytical error because this error has happened before the analysis of that sample. So we can consider it a pre-analytical error. The standard gauge or needle commonly used for routine adult viniculture is, it is 21 to 22 gauge. Okay, hope I have understood. Hope you have understood these MCQ questions which I have shared with you. Please like and subscribe my channel. Now I will take a 5 minutes break, and after that I will resume the next set of 25 questions which are very important for you because these questions are of microbiology. Each and every questions are of microbiology. So I will take 5 minutes break, and then I will share with you the next set of 25 questions. Please share this stream with everyone so that they get also because so that they can also see my live stream and enjoy this live stream. Okay, please share with everyone because sharing is caring. We need to educate everyone. We should not be educated. We have to educate everyone. So here the question is given as which of the following is the primary strain used in Jelen Nielsen acid fast method? So what is the strain which is used in acid fast method? In acid fast training, carbon fusion is used. Okay, carbon fusion strain is used in acid fast staining. And in which bacteria it is used? It is used for Mycobacterium tuberculosis. Now, what is the standard temperature and pressure for routine autoclaving? It is 121 degrees centigrade at 15 psi pressure. Which of the following culture media is selective and differential for Staphylococcus aureus? It is McConquay agar because it helps us to differentiate between lactose fermenting bacteria and non-lactose fermenting bacteria. Here the question is given as the VDRL test is a non-teripronemal test used to screen for. The VDRL test is used for screening of. Do you know why VDRL test is used? VDRL test is used for which thing? It is used for screening syphilis. Okay, syphilis. Now, the next question is which organism characteristically produced a green metallic sheen on using methylene blue EMB agar? EMB agar. EMB agar. EMB agar is used to screen which bacteria? It is used to screen Escherichia coli. Escherichia coli, E. coli. Okay, now the next question. Which of the following immunoglobulin is primarily involved in mucosal immunity and found in secretion? Mucosal immunity. It is IgA. The answer of this will be IgA. An organism that grow optimally at a cold temperature that is 4 degrees centigrade to 20 degrees centigrade is known as 4 degrees centigrade to 20 degree centigrade. So, It will be psychrophile. Organisms which grow optimally at 4 degrees centigrade to 20 degrees centigrade is psycho. It's primarily transmitted via the fecal or oral route. Can you say me the answer of this question? It will be hepatitis. The answer of this question will be hepatitis A. Now, in the gram strain procedure, which chemical act as a mordant? It will be grams iodine. Grams iodine acts as a mordant because it helps in fixation. Okay, because it helps in fixation process. Now, the India ink preparation is used primarily to visualize the thick polysaccharide capsule of. It will be Cryptococcus neofans. The answer of this question will be Cryptococcus neofans. Okay. Now the next question is which bacterial species is a well-known cause of pseudomembranous colitis following broad-spectrum antibiotic therapy? I am skipping this question. Now I will solve it later. Now the next question is which type of hemolysis is characterized by complete clearing of red blood cells around a colony on a blood agar plate? It is beta hemolysis. The answer of this question will be beta hemolysis. Now the next question is the causative agent of Lyme disease. Positive agent of Lyme disease. CA is a positive agent. Uh, it is Borrelia bagdoririferi. Okay, the answer of this question will be option C. Now the next question is, which Plasmodium species is known to cause the most severe and deadly form of malaria in human? It is Plasmodium vivax and Plasmodium falciparum. Okay, these two. Are you enjoying the stream? Are you enjoying? Please let me know and put some comment in the comment box. And please like and subscribe my video so I can bring more videos for you like this. Here the written as the oxidase test is used to detect the presence of cytochrome C oxidase. Which of the following organism is typically positive in oxidase test? So do you know what happens in a oxidase test? In oxidase test, the bacteria releases H2O2, that is hydrogen peroxide, and forms bubbles. So oxidase test is used to detect. I think it will be Escherichia coli. It will be Pseudomonas ericoshita. Okay, it will be number B. Now, here the question is given as, which of the following is an anaerobic spore forming gram positive bacteria that causes lockjaw? What causes lockjaw? Which bacteria causes lockjaw? Which bacteria causes lockjaw? It is the answer of this question will be Clostridium tetani, because it is a anaerobic bacteria. Anaerobic means it can grow in the absence of oxygen, and it forms spores, which is very much terrifying. And it is a gram positive bacilli, means it is gram positive in nature and is rod shaped. Okay, and this may cause lockjaw in our body. Why the Weddell test done? With the Widal test in a serological test historically used to aid in the diagnosis of Widal test. Widal test is done for typhoid fever to find out if someone has typhoid fever. Widal test is used to diagnose typhoid fever. Okay. Which piece of laboratory equipment is commonly used to sterilize inoculating loop and needles via dry heat? It will be hot air oven. For wet heat sterilization, the answer will be autoclave. Anything which is wet heat, that is steaming, it is known as autoclave. Autoclave is used, and for dry heat, hot air oven is used. Okay, you have to remember it. A hanging drop preparation is a classical method used primarily to observe bacterial mortality. Hanging drop method is used to find out the bacterial mortality. Okay, you have to remember which method is used to find out the bacterial mortality. If someone asks you, then you have to say a hanging drop method. Negri bodies in brain tissue. If we find if we find microscopic finding of Negri bodies in brain tissue is indicative of an infection by which virus? Negri bodies in brain. Negri the Negri bodies in brain tissue. If we find under a microscope Negri bodies in brain tissue, then it is the indicative of rabies virus. What is the indicative? It is the indicative of rabies virus. Okay. Now here the question is given as which of the following intestinal nematodes is commonly known as pinworm? तो पिन वर्म यहाँ पर हम लोग को बोला गया है कि ये पिन वर्म का it will be Enterobius vermiculus. What is the scientific name of pinworm? It is Enterobius vermiculus. Anything if the virus name starts with entero, then it means that it will be found in our stomach. Okay. In the catalase test, which reagent is added to the bacterial colony to observe for bubble formation? It is for catalase test for bubble formation. Hydrogen peroxide is used. Mhm. Hydrogen peroxide is used. Okay. Now the question is given as McConkey agar is a differential medium that distinguishes enteric bacteria on the basis to ferment. Means McConkey agar is a differential medium which enables us to distinguish the enteric bacteria, means the bacteria which are present on our stomach on the basis on the ability to ferment lactose. Okay, it is the answer of this question will be lactose. You have to remember it. Here is the here it is given that which bacteria causes UTI. So the UTI causing bacteria is, but here the question is giving different. Here it is given that which organism is globally the most common cause of uncomplicated urinary tract infection, and the right answer of this question will be Escherichia coli, that is E. coli. Now here the question is given as which of the following strain is considered a differential strain used to identify acid fast bacteria such as Mycobacterium tuberculosis, and it is Gel Nielsen strain, acid fast training. Okay, so thank you, friends. Now I will end the live stream here. Please like and subscribe my video if you like it, and please share with everyone so they can get a very. There is a very high. These are very high quality questions of medical lab technology, and it will be very much helpful to you if you are a student of medical lab technology or if you are going to give any competitive examination. So thank you, friends. Now I am going to end the live stream. Okay, thank you.